Hello everyone, let's learn what is somatic cell nuclear transfer, SCNT. It is a laboratory technique used in cloning and stem cell research. The basic mechanism involves transferring the nucleus of a somatic cell, any cell other than a sperm or egg cell, into an enucleated oocyte, an egg cell that has had its own nucleus removed. Here's a step-by-step breakdown of the process. 1. Obtaining the somatic cell and the oocyte. A somatic cell is obtained from the organism to be cloned. These cells can be from various tissues. An oocyte is collected from a female of the same species. 2. Enucleation of the oocyte. The nucleus, which contains the genetic material of the oocyte, is carefully removed. This leaves an enucleated oocyte containing the cytoplasm and other cellular components. 3. Preparing the somatic cell nucleus. The nucleus is isolated from the somatic cell. In some protocols, the cell cycle of the somatic cell is synchronized to a specific phase, often G0 or G1. 2. Enhance reprogramming. 4. Nuclear transfer. The nucleus from the somatic cell is inserted into the enucleated oocyte. This can be done in a couple of ways. Direct injection. The somatic cell nucleus is directly injected into the cytoplasm of the enucleated oocyte using a micropipette. Cell fusion. The entire somatic cell is placed in the perivitelline space, the area between the oocyte's plasma membrane and the zona pellucida, and then fused with the enucleated oocyte using an electrical pulse. 5. Activation. The reconstructed oocyte, now containing the somatic cell nucleus, needs to be stimulated to start dividing as if it has been fertilized by a sperm. This activation can be achieved through various artificial methods, such as electrical pulses. A short electrical shock can trigger cell division. Chemical treatments. Certain chemicals like strontium chloride or calcium ionophores can mimic the signals of fertilization. 6. Reprogramming. Once activated, the cytoplasm of the oocyte contains factors that can reprogram the somatic cell nucleus. This reprogramming process is crucial. It allows the somatic nucleus, which was specialized for a specific cell type, to revert to a totipotent or pluripotent state, capable of directing the development of a whole organism or various cell types. 7. Embryo development. The reprogrammed oocyte begins to divide and develop into an embryo in vitro, in a laboratory dish. If the goal is reproductive cloning, this embryo is allowed to develop to the blastocyst stage, an early stage of embryonic development, and then implanted into the uterus of a surrogate mother to develop into a fetus. If the goal is therapeutic cloning, the blastocyst can be used to derive embryonic stem cells, which are pluripotent and can be differentiated into various cell types for research or potential therapeutic applications.
Thank you.